Hi everyone. Our group has protein as our macromolecule, and my name is Lila Joya.
My name is Sarah Bradford.
I'm Delvinae Beckles.
I'm Kadreen Malcolm.
And I'm Kayla Hicks.
So we're going to go ahead and get started with what proteins are. Proteins are polymers made of amino acid monomers. A chain of amino acids are known as polypeptide chains. These chains can either form a protein or is a part of several polypeptide chains that make up a singular protein. So there's four structures of proteins. It's primary structure, the secondary structure, tertiary structure, and quaternary structure. The first one is a linear sequence of amino acids in one polypeptide chain. The secondary structure is an A helix and B sheet structure that comes from hydrogen bonds between the main peptide groups. The alpha helix and beta sheets are folded to create the tertiary structure that is held together by non-polar covalent bonds and von der Waals forces. And the final structure is multiple folded protein structures that create a subunit complex bonding with non-covalent bonds. And here to the right is a diagram of the different protein structures and what they look like. So to the left, we have the hemoglobin, which is responsible for transporting and carrying out substances throughout the body in the blood or the lymph. And then to the right, this is actually the COVID-19 antibody. Antibodies are responsible for protecting the body from foreign pathogens. So I picked COVID-19 because that's currently what we're going through right now. We're trying to build antibodies to fight off this vaccine and the different variants.
So there are many locations and functions of proteins in cells. The first location is the nucleus, and one of them is chromatin, where the chromatin is made of DNA and proteins. The second is the nucleolus, where proteins are imported from the cytoplasm and are assembled with ribosomal RNA into subunits of ribosomes. Then there's the nuclear envelope, and this proteins help coil the DNA molecules and shorten the length to allow it to fit into the nucleus. Then there's the rough endoplasmic reticulum, and the majority of the proteins found there are glycoproteins. And in the Golgi body, this is where the proteins are transported from the rough ER, where they are then modified. Then there's ribosomes, and ribosomes are made up of RNA and proteins. And there are two types of ribosomes, free ribosomes, and the proteins from the free ribosomes are found in the cytosol. And then bound ribosomes make proteins that insert into membranes for packaging or secretion. And in the mitochondria, enzymes are found in the mitochondrial matrix, and proteins in this location help aid in cellular respiration. In peroxisomes, proteins made in the cytosol and the ER are found here, and enzymes are used to help grow larger and also involved in oxidative reactions. Then we have the cell membrane, and it's a phospholipid bilayer. And membrane proteins, there's two of them, enteral and peripheral, and they can be found on either side of the membrane. And there are many different functions for these proteins, one being attachment to the cytoskeleton and extracellular matrix. Then within the cytoplasm, there's cytoskeleton, and there are three proteins, tubulin and actin, and then keratin is another type of protein. And in the cytoskeleton, proteins are used to maintain cell shape, cell movement, and much more. In the cytosol, enzymes catalyze the first steps of glycolysis or sugar breakdown. And lastly, there's extracellular matrix, and the proteins are glycoproteins. And some examples of these are collagen and fibronectin. There are also proteins in the cell wall and chloroplast, but it's very low numbers.
So next, we're going to talk about keywords. We're going to talk about enzymes, and enzymes are proteins that act as biological catalysts. So they make a biochemical reaction more likely to succeed by lowering the activation energy of the reaction, and they're specific to a substrate. So to the right, we have a photo of enzyme activity, and it just shows a substrate going into an enzyme, and then finally the products being released. Next, we have amino acids, and those are monomers that make up proteins. And the structure of an amino acid is an alpha carbon bonded to an amino group, a carboxyl group, and a hydrogen atom, which is also shown in the photo in the right. So next, we have a polypeptide chain. And a polypeptide chain is made up of amino acids that are linked together. So basically, amino acids are the building blocks of polypeptides, and polypeptides are the building blocks of proteins. Next, we have a peptide bond, which is a chemical bond that is formed between two molecules when the carboxyl group of one molecule reacts with the amino group of another molecule. Within a protein, amino acids are linked together by peptide bonds. And then we have a proteome, which is the entire complement of proteins that can be expressed by a cell, tissue, or organs. Lastly, we have the structure of proteins. So like Sarah mentioned earlier, there are 4 structures of proteins: primary, secondary, tertiary, and quaternary. And the primary is the simplest level of protein structure. It's the sequence of amino acids in a polypeptide chain. Secondary refers to local folded structures that form within a polypeptide due to interactions between atoms of the backbone. Tertiary is the overall structure of a polypeptide, and it is primarily due to interactions between the R groups of the amino acids that make up the protein. And quaternary is when multiple polypeptide chains come together and they form the quaternary structure of the protein.
The common way to test for protein is through the BIRA test. And the BIRA test is the BIRA test, it tests for compounds with 2 or more peptide bonds, and protein has multiple peptide bonds through its links with amino acids. It is formed between the alpha amino group of one amino acid and the alpha carboxyl group of another amino acid with the removal of water, and this is called condensation. The BIRA reagent, which is used for BIRA testing, is composed of sodium hydroxide or potassium hydroxide with hydrated copper sulfate and potassium sodium tartrate, which is used to test for a solution for a protein and the color change is observed. If protein is positive, then it will become a purple color, and if protein is negative, there will be a blue color as shown in the photos to your right.
Another detection technique for detecting proteins is the ELISA method. ELISA stands for enzyme-linked immunosorbent assay, and it's a detection and quantification of soluble substances like peptides, proteins, hormones, antibodies, and more. So the major principle behind ELISAs are antigen-antibody interactions, and there's a variety of applications of ELISAs in cancer screening, drug testing, pregnancy testing, and more. So on the picture, we see a probably 96 well plate. So you use a micro well plate and you load your samples in, and we can use the indirect ELISA as an example. So let's say you're trying to test for the presence of a breast cancer resistance protein, which is also known as BCRP. You basically order a BCRP ELISA from a manufacturer, and the antigen for BCRP will be stuck down at the bottom of the plate. So in all these examples, the green dot would be the antigen, and then in sandwich ELISA, it's basically the pink antibody. So you get a BCRP ELISA, and then you would load your sample into the plates, and your sample would have BCR proteins, or it could be a mixture of proteins. But in order to isolate the BCRP, you'd load your sample, and theoretically, your BCRP should bind to the antibodies of the BCRP should bind to the antigen that's stuck at the bottom. So after that, you'd wash your samples, and then you would load a secondary antibody, and that would bind to the BCRP, and it would have to be specific for the BCRP. And that secondary antibody would have an enzyme linked to it, and then you'd wash it, and then you would load a substrate that is specific for that enzyme on the secondary antibody. And basically, once you load that substrate for the enzyme, you'd get a change in color. So The ELISA can detect and quantify the presence of protein because it's very specific and uses antigen antibody interactions, and then it's colorimetric. So the darker the color, the more protein. So this is just a good method for protein detection, and it's used a lot in a lot of clinical research. So we thought this would be interesting. And these are our citations, and we just want to say thank you for watching our presentation.